So our next speaker is Dr. Paulyn Cartwright. Dr. Cartwright is a professor in the Ecology and Evolutionary Biology Department at KU. She received her BS in biology from UCLA in 1990 and a PhD from Yale in 1997. Currently, she heads the Cartwright Lab, which takes an integrative approach to understanding Medusozoan evolution by using a variety of approaches, including phylogenetics, genomics, and development. Specifically, her lab is building a phylogenetic framework that can be used to guide future studies of Medusozoan development. And today she will be giving a presentation on genome editing in the age of CRISPR. Let's welcome Dr. Paulyn Cartwright.
Thank you, Jake, for the introduction, and thanks to Holly for your organization and for thinking of me and inviting me here. It's a pleasure to be here today. As Jake said, I'm a professor in ecology and evolutionary biology. I'm actually a colleague of Chris Beard, so we're from the same department. And for the last dozen years or so, I teach undergraduate evolutionary biology. And one of the nice things about biology majors in Kansas is that almost every single major has to take evolution. So I've had pretty much every single biology major in the last decade come through my class, which is nice, except when you go to the doctor's office now. All the doctors and all the nurses and everyone, hey, Dr. Cartwright. So anyway. And then I teach a research-intensive course on, it's called Evolution of Development, and that's really geared towards seniors. And in my lab, I mentor grad students and undergrads, and as Jake said, we're really interested in the evolution of jellyfish, unraveling their evolutionary path. Medusozoans are basically a fancy name for jellyfish and their relatives, and unraveling their patterns of evolution, but really I'm interested in understanding the genetic mechanisms underlying all this diversity. So you may be wondering, like, why am I talking about CRISPR? How does a jellyfish evolutionary biologist get interested in something like CRISPR? And I will talk about the application of CRISPR to my research towards the end of my talk. But I really think the question is, why isn't every biologist now interested in CRISPR and jumping on the CRISPR bandwagon? It's really an exciting technological innovation that has occurred in the last really 5 years. One of the most exciting ones that I can remember in my adult life. And I was thinking back about what are the technological innovations that's really changed the way that we approach our research. And what I can remember in my career would be like PCR, the polymerase chain reaction, where you can target specific pieces of DNA and amplify it. Another innovation that I can remember is the advent of what's called next generation sequencing, where we can sequence whole genomes relatively cheaply and assemble them and compare them. And these really did change the way we think about both our approaches to research and how we understand biology and biological diversity. And the next major one, I think, is CRISPR. And CRISPR is just a way to cheaply and relatively easily, but more importantly, very specifically, alter DNA. Like a gene of interest, we can now mutate, take out, replace with another gene, and we do this in a very specific and relatively easy way. Unlike the other technological innovations that I've talked about, like PCR and genome sequencing, this one is a little bit different in the sense that it has the potential to affect the very essence of the genetic content of the species you're working on, including humans. And when it can affect the genetic content, what I mean by that is it can be passed on through the generations, so that we can manipulate genes and gene content now such that it effectively can affect our evolution, both our own species and other species, because it's inherited through the generation. And why is this different from other treatments that we talk about, like gene therapy for cancer, other types of gene editing techniques? And the reason why is because CRISPR works on the embryo. It works when it's a single-celled organism, whether it be humans or jellyfish. And when you're modifying DNA at a single cell instead of treating an adult that has disease, even with genetic techniques, when you're modifying at a single cell, which means that newly modified DNA is in every cell of the body, including the sperm and the eggs, which means that that modified DNA is passed on through the generations. And that's the distinction between how we genetically modify genomes now and how we would use genetic techniques to, say, treat diseases in adults in the past. But this isn't just a few disease cells. We're really altering every cell in the body at the embryonic stage. So those are the implications, and that's why there's a lot of talk about it. But I'm a scientist, and I'm going to really just talk to you about the science for now and then open it up to questions. I will say, I don't think I have an hour's worth of slides, so if you want to interrupt during, please feel free. Let's have this be, casual, and if you have any questions as I'm going through, please feel free and raise your hand. But the first question is, what is CRISPR? And I'm not even going to say it. It's just this long scientific term, short for this. I was hoping to have a pointer, but I think it ran out of batteries on me. Hold on. Okay. Well, it works here, but it doesn't work there. I think it's not bright enough. Okay, anyway, I'll just point. So, like most biotechnology innovations, humans did not invent CRISPR de novo. What us humans did, and I mean us scientists, is made an observation in nature, and then we're able to modify it for our own purposes. We're not smart enough to come up with something so amazing as CRISPR. This was borrowed from bacteria. And what CRISPR is in nature, which was observed and characterized in nature, is an immune system. It's part of their immune system of bacteria. And much like our own immune system, it's a learned immune system. So bacteria are plagued by viruses. See that little spaceship-looking creature at the top? That's a virus. And viruses can't replicate on their own. Oh, thank you. Thank you. So here's this virus, and it can't replicate on its own. What it does, no virus can replicate on its own. It requires other people's cells, whether it's a multicellular organism like ourselves or a bacteria. But in this bacteria, it injects its DNA, shown here in yellow. And then, under a normal virus life cycle, it incorporates its DNA in the genome of the bacteria. It borrows the bacteria's machinery to replicate its DNA, and then it reassembles viruses, and the bacterial cell busts open. Well, bacteria don't like that, obviously, to be hijacked by this virus, so they develop their own immunity against viruses. So this is what they do. It's like a vaccination for us and our immune system. When the virus, the first time a bacterial cell is exposed, it injects its DNA, and then the bacteria incorporates a small fragment of the virus DNA into its genome. And then the next virus, which would be green, would be here, and another kind of virus would be here, such that in the course of this bacteria's lifetime, when they're exposed to different types of viruses, they incorporate these tiny little bits of their genomes, such that, so this is like the vaccination stage, and then this is the immune stage, such that the next time a virus, this same virus, injects its DNA, what happens is that little piece is transcribed, and there's a little spacer that's made with it, meaning that you take this little piece of DNA and you make an RNA. A little piece of RNA that's an exact copy of the small virus and this little spacer that forms this little hairpin. Okay, then what happens is this protein called Cas or Cas9 recognizes this piece of RNA, binds to it, and together they work to cut up the virus DNA. So they chop it into little bits, they protect the cell for the virus invading the genome, replicating, and ultimately busting open the bacterial cell. So this is the way bacteria, this is an immune system that bacteria evolved to protect themselves from viral infections. They have tiny little pieces of DNA from previous exposure and these spacers. Well, why are we interested in that? Well, because of this remarkable observation that a tiny little piece of DNA, and we're talking like 20 bases of A, C's, G's, and T's, can so specifically recognize another piece of DNA and in combination with a protein, cut it in a very specific way. 20 bases is all we need with protein to cut DNA in a very specific way. And again, humans are not smart enough to invent something like this. This comes from a bacterial system. But what humans did, and in particular these two human scientists, Jennifer Doudna and Emmanuel Charpentier from Max Planck, is they made this observation that we know that we can now harness CRISPR as the natural bacteria immune system to cut any DNA of interest in a very specific way that we want to. We have a gene of interest, we want to mutate it. All we have to do is make a tiny little piece of RNA that matches that DNA of interest, that matches it, and then make sure we put this little tail on, this little hairpin, mix it up with a protein called Cas9, and then we can cut a DNA. And we can do this inside cells, living cells. And so that's what they developed as a system with which we can now put this cocktail of RNA and protein and cut up DNA in a living organism in a very specific way. Whatever gene we want, let's just mutate it. Are there any questions? Okay, so what we do now, me as a biologist, have a gene of interest. I tell a biotech company, these are my 19 bases, A, G, 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 T, C, A, G. Those are the bases I want to target because they match my gene of interest. Add this little hairpin to it, please, and send it to me in a tube, and the next day I get it. That's in reality what we do. And then we take the Cas9, which was originally isolated from bacteria, but now they cloned it and engineered it. These biotech companies will send me a tube of that. I mix them up. I mix this and this. I put it in a little needle, and here are my embryos. And I inject them. And now I can mutate any gene I want of interest. And it really is that easy. I have undergraduates doing this. It's a very simple thing. Find the gene you're interested in, inject it, see what kind of mutation, grow it up and see if it looks strange or if it affects a certain cell process or whatnot. Did I skip one? Okay. So there's a couple of different applications now that we can do for this. So here's our bases of interest. We make sure we add this little hairpin. Why do we have to add this little hairpin? Well, just for whatever reason, the protein needs to recognize its structure to bind to it, this Cas9 protein. Once it binds to the DNA, because it matches the bases, it finds it, it cuts it literally like scissors, and now we have a mutated gene. Well, what do we do with it then? There's a couple things you can do with it. Maybe we want to see if this gene is essential to a particular function. So just mutate it and try to grow up your embryos and see if they die or if they look weird or it changes something. Maybe the whole gene, because of a prior mutation, is deleterious to the animal. Cut the gene out. Cut it here and cut it there. Just take out the bad stuff. That's another application. Or maybe we want to cut out a bad gene that's undergone a mutation and put in a new gene, a new sequence that doesn't have the mutation. So in our cocktail, let me back up. In this cocktail that I put here in the needle, not only do I add my piece of RNA and my protein, but I can add a piece of DNA that's the good DNA. Inject it in, The CRISPR-Cas9 cuts out the bad DNA, and because they have good DNA in there, it will be replaced. And all eukaryotic cells have a natural mechanism to repair broken DNA. So if they see things that are cut, they go in and they sew it up. And so if it's cut and there's a new piece of DNA that fits in, it'll automatically sew it up again. So that would be called a transgene or transgenics. Take a gene from somewhere else, put it in a new organism. Or this would just be called a mutant or a knockout. Go, Jake.
Yeah, so what's the enzyme that's repairing the gene, like inserting that new gene when you just have the gene?
There's all kinds. What?
Is it like a polymerase or a ligase?
Yeah, they're always there. It depends. So if you're missing bases, DNA polymerases will come in and fill some in, right? If you're missing one versus the other. But ligases, you remember your biology, good for you. Ligase is definitely the glue. The ligase will literally glue these two pieces back together, these two bases. But you will have some polymerases sometimes filling things in. So both will work. And those are just normal mechanisms. We don't even worry about that. We just know it's going to happen. But what you do is you bombard it with this, what we call donor DNA. So there's so much in there that they almost like accidentally take it up at the cut site and then repair it. Okay, so there's so many different applications to CRISPR. When I was asked to give this talk, I honestly didn't know where to start. I'm like, what should I talk about? What is the most interesting? And so instead what I did is I Googled it, and I Googled Google News, and I looked at all the science news, and then I sorted it from most recent to, whatever. And I literally had dozens of examples of published papers that had made in the science news through March. So in the last 6 weeks. So I thought, okay, well that's what I'm just going to sort through the last 6 weeks and see what cool applications to CRISPR have been published and have made it to the science news literally in the last 6 weeks. So one can imagine if you look back in the last 5 years, all the different applications to CRISPR. So the examples I want to talk about now are not being applied to humans currently. They're basic research being done either on cell culture or on model organisms such as mice in the hopes that eventually they can be applied to humans or plants or whatever. So here's one I found, which I thought was really cool in the news, and that is we know there's a lot of blood disorders that are caused by mutations in a single gene, hemoglobin. So this would be sickle cell anemia, beta thalassemia, and the other thing that we know through basic research is that embryos and fetuses have a different type of hemoglobin coded by a different gene than adults. So there are different genes coding depending on your life stage. Also that's been observed from scientists is that sometimes there's a naturally occurring mutation where the fetal hemoglobin doesn't turn off and continues to function in the adult. And so some creative scientists said, okay, well, what if we recapitulate that naturally occurring mutation in patients with sickle cell anemia, such that the fetal one can now be expressed in the adult when the adult one is not functioning that well. So what they did in cell culture is they went and they used CRISPR to target and mutate the fetal hemoglobin gene such that it doesn't turn off in the adults. And they did see that indeed it expressed and it compensated for the mutated adult hemoglobin in the hopes to mitigate some of the symptoms. Now, just keep in mind to apply this for disease effectively, I would think, and I'm not a doctor, I would think one would need to do it at the embryonic stage, meaning you're at risk for sickle cell anemia, screen the embryos, and mutate the fetal protein. I mean, obviously another way to do it would be in stem cells for blood and inject the patient, but that probably would be an ephemeral effect if it weren't actually in the genome of the body. But that's one approach for using CRISPR for disease. Here's another one I found March 12th, and this was a study that was I think it was published in the Proceedings of the National Academy of Science, which they wrote about it, the Science News, where they, so ALS is due, they know, to a protein of a single gene, shown here. And what it is it's overexpressed and it wreaks havoc on certain aspects of the nervous system. So what they did here is, using basic research, they looked at all the different genes that interact with this particular gene. And they literally took CRISPR and they said, oh, we think there are 200 genes in this pathway that interact with it. So let's mutate these 200 genes one at a time and see if any of them affect the overexpression of this gene to help mitigate the symptoms in cell culture. So a lot of talk has been about using CRISPR on mosquitoes because mosquitoes carry lots and lots of horrible diseases that affect humans, such as malaria. In this particular study that just came out in the last 6 weeks, they used CRISPR in embryos, in mosquito embryos in the lab, to make to mutate the embryos such that they are resistant to the parasite. So that these particular mosquitoes don't carry the parasite, which then when they bite humans, they don't transfer the parasite to the humans. This is a little more palatable for some than previous efforts, which have been used to use CRISPR to make the mosquito sterile. Because when you release the sterile mosquitoes into the environment in conjunction with another tool, which is called gene drive, so you modify mosquitoes such that they're sterile. And usually when you modify a single gene, according to Mendelian inheritance, only half the offspring have them. So you do a bunch in the lab, you release them in nature, and they're really just a small percentage. But there's another technique, I'm not going to get into the details of it, called gene drive, which basically tricks Mendelian inheritance such that every or most of the offspring carrying that modified gene inherit it. So you can take a few hundred individuals that you modify in the lab, release them out to nature, and it can sweep through the population in a few generations because you're doubling it every time. All the offspring will have the modified gene. And they have done this with some success. I think it was in Brazil, and they definitely did it in Florida for Zika. So they released the mosquitoes, caused sterilization to spread throughout the population, and brought down the population of disease-carrying mosquitoes. But it brought down the population of all mosquitoes. Some people are concerned with the implication that I try to think why we care about mosquitoes, and if they were gone, would we really miss them? Would there be anything? So obviously there are concerns of unknown environmental implications to this, but as far as I can think of, I can't think of any reason why we would miss a mosquito. But so those are a couple of approaches that they've used for CRISPR in conjunction with this cool tool called gene drive. As far as I know, gene drive only works in some insects, but I know that it's being researched more. Here's another one I found, which I found very interesting, and I read up on this quite a bit, and that is to use CRISPR in what is called gene therapy. So everything I've been talking about is applying it to embryos so that all of them have it. Gene therapy is to apply it to adults with a disease, like leukemia, for example. Gene therapy has been used. And the old way in which these experiments have been done in humans with gene therapy is you take a virus carrying a good gene, you inject it into the blood cells, for example, if it's leukemia, and the virus incorporates randomly into the genome of these cells, carrying the good gene, and then the good gene hopefully outcompetes the bad gene or mitigates the effects of the disease. If you genetically engineer it such that now CRISPR is in there, it means that you can target exactly where this virus goes in the patient's genome. It's not just some random incorporation. So ultimately, you can use it to replace the bad gene in the cells. But as I said before, it's going to be very localized and probably the effects ephemeral because you're injecting a subset of cells in the adult that are genetically modified. You're not actually changing all the genes of that particular adult. But it could be effective for certain like blood diseases, I think is what they're thinking this approach. So it's basically, gene therapies are very controversial, and again, in large part because it's so non-specific. You're letting a virus wreak havoc on your genome and you don't know where it's inserting. If you apply the CRISPR technology, you can say, only insert here where the bad gene is, and then you can more readily control the effects of that. But again, it's on adult patients, so it's not like it's going to get passed on to the next generation. It's not like it's affecting all of the cells. So I think time will tell whether or not this approach on adult cells becomes effective. This one I just loved. Turns out, we all like hoppy beer. IPAs are very hoppy and trendy. But hops, which I didn't know, are both expensive and hard to come by. Turns out there's a huge shortage of hops because of this increase in demand for hoppy beer. So, I'm glad none of my grad students are here. They're like, why can't we do this in the lab? Because these young students thought, we know a gene that confers the bitterness of hops. So let's take yeast, which are brewer's yeast, which are single-celled organisms, using CRISPR, stick that gene into yeast, and then ferment yeast instead of having to rely on hops. So sure enough, they did it. They genetically modified, lucky students, I'm sure all grad students are so jealous that they get to work on beer. They genetically modified yeast and produced beer with a hoppy flavor. So they're probably going to get really rich, or I should say UC Berkeley's going to get really rich off this, I suspect. And here was a study in which, again, they used it on adults, not on embryos, to treat patients with dementia. And the reason why I thought this was an interesting study of the dozens and dozens I went through in the last 6 weeks is because another reason why CRISPR is controversial is because it modifies the DNA. And that's scary, and it mutates it. They modified the Cas9 protein, so it doesn't modify the DNA. It actually just modifies the expressed RNA, which is a very temporary thing. So the DNA will stay mutated, but you'll fix the intermediary step, the RNA, in a very temporary way. I didn't know you could do this. I just learned it from reading the original paper here. It seems very promising, again, but a very ephemeral treatment. They called it, instead of Cas9, it was like Cas-RX, which it's mainly like a drug. That they can use it like a drug to temporarily treat a disease at the RNA level and not permanently modify the DNA. And again, I think as treatments, experimental treatments for humans, I think doing that instead of getting into the genome is a lot more palatable for some. And then I'm just going to show a couple more examples. This one is using CRISPR at embryos, so one would have to screen embryos that were at risk for Fragile X. And turns out Fragile X syndrome really has to do with just a single gene, and there's these repeats in this single gene that cause it. So they successfully, in culture, used CRISPR to take out that mutated part of the gene and show that the cells were happy and healthy in culture. Okay, this one I just loved. So silkworms are a big industry in China, produce silk, but in silkworm cultures, they're plagued with viruses. And every time they're plagued with viruses, the whole silk industry takes a plunge. And these scientists said, well, wait a minute. If bacteria can fight off viruses in nature using CRISPR, why can't we put the whole damn CRISPR system that bacteria has into our silkworms so that they too can fight off the viruses? And they did, successfully. So now they borrow the immune system from the bacteria, put it into embryos of the silkworms, and now have these silkworm lines that have the same type of immunity to the viruses that the bacteria have had all along. I thought that was a really cool application. And then I just show this because, and this wasn't a study, but I show this because CRISPR has been a lot slower to take hold in plants, and in particular crop species, than in animals. And the reason why this is the case is two reasons. They have pretty good ways to transfer transgenes in genetically modified crops already. They have these various viral systems that are pretty effective at doing it. So there has been less motivation. That's why we have much more genetically modified crops than we do genetically modified animals. Because prior to CRISPR, it was really easier to do it due to the different types of technology that they developed in crop species. So there was less motivation, but everyone has CRISPR envy, including Monsanto, in part because it's cheaper to do it CRISPR, in part because it still is more specific than the ways that they do. But the challenge to apply CRISPR to plants is the delivery system. Animal embryos tend to be very soft, and you can inject a lot of them and they readily take up DNA. Now we're dealing with seeds or pollen, and getting those into them has been a bit more of a challenge. But I just show this to say that Monsanto thinks that there's a future and is investing heavily in developing CRISPR technology for crop species. And in fact, last week, USDA said, well, because CRISPR is so specific, and because we know exactly what it's doing, we're not even going to regulate it at all, because we believe in this technology. So that was a decision made by the USDA. But as far as I know, I don't think that any are in the market, or I actually don't even think they're that close, because it is more challenging in plants. Okay. I can stop here. Is there any questions or applications? Sure.
Well, ultimately, is this kind of part and parcel of on the road to designer babies?
Oh, yeah. I don't know. Is there technology to screen embryos for traits that we know about and replace the genes? Yes. Yes, there is.
So what about the ethics of this?
Yeah, I know. So, okay, number one, I will say that there's a lot of traits that are multigenic, or that most traits are, like, intelligence, whatever that means. So, no, because they're much more complex traits and they're not single gene, and that's just not going to happen. Of the few that are single gene that we know about and are well characterized, can that happen within our own technology? Yes, absolutely. So, yes. Yeah.
It seems a trio of studies have shown that Cas9 has an intrinsic RNA lysing ability. A bacterium would express Cas9 in response to an RNA viral challenge, and it could kill the RNA virus in some cases, although the virus can respond by making an anti-Cas9 protein to defend itself. Does this open any possibilities for new applications, or conversely, does it open avenues for unanticipated side effects if we wind up messing with RNA inadvertently?
Right. So, in bacteria, there is an evolutionary arms race in the bacteria and virus infections. That bacteria have evolved this Cas9 system to fight them off, and in response, viruses are evolving ways to evade the Cas9 system, let's say. And in response to them, bacteria have evolved other Cas systems that are slightly different than the Cas9 that can evade the evaders. So this arms race has gone on, and it's been well documented. And it's been good for us as scientists, because then we've been able to isolate the other kinds of Cas proteins that do slightly different functions and be able to tweak with that. I think your question is, if we release this in nature, won't there be the same responses, or won't there be other unanticipated responses to evade this Cas9? So I have, in terms of the science, I'll tell you that most of us who are doing CRISPR are not genetically engineering the actual CRISPR system. So I showed you the silkworms where absolutely, now they're producing their own Cas9, their own RNAs, and they're probably, due to selection, because evolution happens, will develop other mechanisms, to counter this. What we do is we just use that system and inject it artificially to modify other genes. So the Cas9 itself, I don't anticipate will evolve from how we do it. But I think there are lots of unknown effects to putting these modified genes in nature that we definitely can't anticipate. Like with the mosquitoes. People are afraid. I'm kind of excited about it. So. Okay, so I'm a scientist and a researcher, and as you might know, we love to talk about our own research. And you all might be interested in CRISPR and the effects to human health and societal implications. I'm really interested in jellyfish evolution and what I can do to learn about that. So you're stuck here, and you're gonna, for the rest of the time, I'm gonna talk about my own research. So, okay. So I work on, it's this group called cnidarians, and it's really jellyfish and their relatives. And their relatives are sea anemones, coral, soft coral, sea pens, and box jellies, stalk jellies, and this group called hydrozoans. Those that have a jellyfish stage to their life cycle are in a larger group called medusozoans. So this is really the group that I focus on in my lab. And what I'm interested in as an evolutionary biologist is in how this diversity of forms arose at the DNA level and at the genetic level. So what was responsible to get all these different things, and particularly all these different amazing life cycles? Within this group, the hydrozoans, their life cycle starts with like a benthic stage. That's a lot like a coral. They have these polyps all interconnected. And then they alternate with a pelagic jellyfish stage. And so that has been the focus of really since graduate school. The focus of my study is understanding these life cycles, how they evolved. And in hydrozoan evolution, many times this jellyfish stage has been lost in some species and then re-evolved again. So that, and so I've used phylogenetics to reconstruct this pattern of evolution, and now I am interested in understanding the molecular mechanisms. And I can say at no time since I've been doing this, I have to say 25 years, half my life I've been interested in this question, have I been more excited about where this research can go. And the sole reason for this is because of CRISPR. So I'm going to explain to you what we're doing. First, I want to introduce you to my favorite species. This is called Podocarina. In nature, it lives on the shells of hermit crabs. It lives on the gastropod shells that are inhabited by hermit crabs. And that's the only place you'll find it. And you'll only find it on one species of hermit crab that's inhabited by one species of gastropod shell. Sometimes they're called, I forgot what the common name is, like fuzzy crabs or something like that. Furry crabs, I think. And you can see that they look furry in the intertidal. The nice thing about this group of hydrozoans is you can take a little piece of it and tie it onto a microscope slide, and they do just fine in growing on microscope slides. And I put them in microscope racks and little tanks, and I can culture them in my lab in Kansas, and they're perfectly happy. They don't even know they're in Kansas. They don't even care. So I fooled them. The other thing is that, which I love about Podocarina, is here are the polyps, but then they will bud off the side of their polyps little jellyfish. Eventually these jellyfish will be released and they swim in the water column, or in a bowl that I transfer them to in the lab. And then these become reproductive And there's either males or females, and they release sperm and eggs, so that I get a ton of embryos. And that's, again, is why I'm so excited about CRISPR technology. For those of us that aren't interested, or not uninterested, for those of us that don't work on human disease and human health and are more interested in other organisms, we can apply CRISPR to this as well. The only criterion so far that we need to do it successfully is access to embryos. And the more you have, the better, because you just have more chances of injecting them and having it work. And so I like to brag to all my colleagues who study worms and other things. Ha ha, too bad you don't get the embryos that I do. I get like hundreds a day. Don't you wish you worked on my guys? So, because I do CRISPR and you can't, not yet. But I'm sure it'll get better. So we're one of the early adopters, meaning me and the 2 other labs that are doing this on cnidarians, of CRISPR in invertebrates, and partially because we can access embryos. So I want to show you the life cycle of Podocrina. It grows on a hermit crab shell or a microscope slide. It doesn't seem to care. It releases these jellyfish. You can't see them by the naked eye. They're tiny. Or, I mean, I'm used to looking at them in a bowl, but you really need a microscope to really see the details. They'll release either eggs or sperm. They undergo external fertilization. They make this little larvae, swimming larvae. And then the larvae sits on its head and metamorphoses to a polyp. The polyp then grows out like this and asexually buds other polyps. And that's the whole life cycle. And we can easily do this in the lab. And I've had the same strains for decades. So, so that's Podocrina. Here's another species I have in the lab. And this is called Hydractinia. It's the closest relative that we know of to Podocrina. It also grows on hermit crab shells. It also is happy in the lab. The main difference is it doesn't have this jellyfish stage. It lost it in evolution. Instead, it has these little gonophores that make eggs or sperm, which then are released from the colony itself. So, here's Podocrina with the jellyfish stage. Hydractinia doesn't have it. What happens is like it starts to develop a jellyfish, but it stops. It's truncated, and then it makes eggs or sperm, and they're released. But other than that, they're very, very similar. And that really has been, for, gosh, it's so sad. For 25 years, I've been really interested in this evolutionary transition, and that's been the focus of my research. And so I'll show you what I've been doing prior to CRISPR. So I've been isolating genes that are specific to jellyfish development and that are not there or not expressed in things that lack a jellyfish. And I mainly use these 2 species, but I scan the whole phylum, basically, looking for genes that seem to be important in jellyfish development or specific to them. So in blue shows the development, these stages are the development at different stages. Because see, you get different stages of medusae development in the microscope slide. So you can just fix them at these different stages of jellyfish development. And then where blue is where this gene is operating. So this is like with my former student, Steve Sanders, we've been looking at things that are specific and that are either the gene is not found in Hydractinia or it's definitely not expressed in jellyfish development. And we use lots of genomic techniques to do this, and we have a handful. So these are all the genes. A couple of these, like these Hobbit's genes, are missing from the genome altogether, and its sister taxon, the closest relative. And others are just specific to jellyfish development. So we have all these genes using genomic techniques that we now can show are needed for jellyfish development, but are not needed to make gametes or things in Hydractinia. In fact, they're perfectly fine. So what we're doing now, and I don't have the answer, so I don't want to get all this build up and then have you disappointed. But this is ongoing. We are asking the question, could loss of jellyfish genes, like the ones I just showed you, that it really have taken me almost 20 years to isolate and characterize and find, could loss of these jellyfish genes be responsible for the evolutionary loss of the jellyfish life cycle stage? So I'll say that again, hold on. we have all these jellyfish-specific genes. In the evolution of Hydractidia, jellyfish are ancestral. The ancestor had them. In the evolution of the lineage leading to Hydractidia, was there mutations in one or more of those jellyfish-specific genes which caused them to lose it and therefore lose this life cycle stage? And I think I mentioned before that in the evolution of this group, this had happened multiple independent times. So we would hypothesize that they would be mutations, and every time it happened, but not necessarily the same ones. Different stages of the pathway, different genes that affect Medusae development. So that's the question, that's the hypothesis. And then, so what happened when we were asking these questions over and over, and I wasn't really able to answer them because I didn't have the tools, what happened is that I'm so lucky because the Stowers Institute is in Kansas City, so yay, we have some Stowers fans. Good. I love the Stowers too. We can talk about the Stowers and how much I love it. Okay, so it's down the road in Kansas City, and there is a lab there that works on a sea anemone. And this lab was the first, I want to say the first to successfully use CRISPR in a non-mammalian system. And this happened to be a sea anemone, which is related to the group I study. So what they did in this lovely paper is they took the sea anemone, which naturally has a red fluorescent protein right here. And as a trick, just to see if they can take out that gene and put in a new gene using CRISPR, they use some donor DNA for a green fluorescent protein. They use guide RNAs here and here to cut out the native gene. They put it all in a cocktail, so you have donor, you have guide RNAs that cut here and that cut here. You have Cas9 protein, you stick it in your needle and you inject the embryos of anemones and you grow them up and see what happens. Did they really care about these red proteins here? No. What they were showing is, does the technique work? And this is a nice marker to see if we can use CRISPR in sea anemones. And sure enough, the red would get knocked out and you'd only have green stripes. And it took a couple of generations because the CRISPR didn't incorporate into every single cell, and therefore not every single sperm or egg had them. So they kept having to rebreed them until a CRISPRed sperm and a CRISPRed egg fertilized, and so you would get these what are called mosaics, part red, part green, and eventually you got all green. And it was really nice, and I said, oh, it came out in December. It's like Christmas tree anemones. They did this on purpose, doing red and green. So that was in 2014, and I was trying to figure out what to do with the sabbatical, and it's 40 miles away. So I took my sabbatical, and I learned how to do all these CRISPR techniques in anemones. And again, at the time, this is like we're talking 3 years ago, no one else was doing them in non-mammalian systems. And I was just lucky to have these collaborators at the Stowers down the road. So I learned how to do it. I successfully did these cute tricks in anemones. And then, so this is where we are now. I went back to my question, my original, anemones are cute and fine, but they don't have those really cool life cycles, and they don't have a jellyfish stage. But these are closely related, so I'm going to use the exact same technology to apply to my organisms and my questions. So I took, I wanted to ask the question, this is what okay, what am I doing? Sorry. Could you knock out some of those jellyfish-specific genes that I took 20 years to find and characterize? And literally make a podocarina into a hydractinia, recapitulate that evolutionary loss? Or even more exciting, could we take a jellyfish-specific gene from podocarina, insert it into hydractinia, and make a new Medusae? So, and again, I don't have the answer yet. But that's where we're going. This is why I'm saying I'm the most excited about where my research is going than I've ever been because of this technology. And I'll tell you why I think we can do it. So again, my former student Steve Sanders, who has helped me isolate a lot of these, he's been really working on this technology with me to apply it to hydractinia. And again, you just take a cocktail of your gene of interest, this little guide RNA, this protein, and you inject embryos. And all we do is in the morning, we turn on the lights, and we get hundreds of them. There's just something about day light cycles that they just spawn. They just spawn like crazy. So, but we have about an hour to do this, because once they start dividing, then they're multicellular, and chaos ensues. So we fertilize them, we inject them, and then we grow them up. And because we're adding, like the Stowers lab did, we're adding a fluorescent protein just to check for the technique. We're looking to see if they have fluorescence. So here's the fluorescent protein. This gene doesn't even matter. I mean, we have lots of candidate genes we're interested in, but we wanted one that's just easy to see and express, like the Stowers people that you can easily screen. And so just to test to see if it works. Okay, so here it is. Here's our fluorescent protein. Here's our target gene. We made guide RNAs here and here. We insert this. Here are the embryos. So they're starting to express, but you can't see it, but not all of them are expressing it. They're mosaic. So some of the cells as they were dividing took it up and some didn't. You can't really see, you can't really see it, but this is the larvae. And some of them have the cells and some of them don't. Steve did the exact same thing, as he just bred them for a few generations to make sure that instead of mosaics, that one sperm and one egg that had them have all of them. And this, I don't know what happened to the color, but it's all green. Every single cell is expressing the transgenic hydractinia. So I'm super excited. Now I have to convince the National Science Foundation to give me money, because I'm out of money, because I took a lot of time to do this. And so once I do, then that's exactly what we're going to do. So we're putting in all these Medusa-specific genes in hydractinia. We haven't yet shown that we can knock out genes in podocrinum, partly because these eggs are a lot smaller, because they have to be carried within these tiny little Medusae. So it's a little bit more challenging, but I'm very hopeful that this will happen very soon. But we definitely are starting right away at putting some of those genes in. So, okay, so anyway, that's all I have to say today. I'm opening it up to questions. I have one in the back, and then.
I have two questions. First is a personal question. What compels a person to grow up and want to study jellyfish?
Right.
And the second one is, do you see, for 20 years you said you studied jellyfish genetics, and now you have this technology. Could you comment on the exponential data that will be coming out of this? And does the technology continue to change, that we become better at CRISPR technology as we become more sophisticated and more skilled with the technique?
Okay, so I think there are three questions. Number one, these are these little microscopic things that no one can even see, and yet I've devoted my whole career to them. So I'm not from Kansas, so I grew up in San Diego. So I love the intertidal, and I spent a lot of time. I'm one of those childhood naturalists that grew up to be a biologist. So, but I do remember, I was interested in two things when I was an undergrad. I was interested in evolution, and I just loved any excuse to go underwater, really. And that really was my only true criteria, and it wasn't any more sophisticated than that. I found a lab for graduate school that would take me, that would fulfill those things, and they worked on hydractinia. And, but this specific question about life cycle evolution is something that I just really started to get interested in as I started unraveling the patterns of evolution within this group, because it was remarkable to me that you have this complex character, a jellyfish, that can get lost and re-evolve multiple times in the hundreds of million years that they evolved. And I was really interested in those types of patterns. So that's what got me into it. So CRISPR, is it going to be exponential? And I'm going to tell you it will if we can convince the funding agencies that it's worth it. It will, and that's what I'm trying to tell them as well. And so, yes, because that transgenic that I showed you was developed in November, and since then, Steve went on to another lab, and he's in Pittsburgh. He left me, but we're still collaborating. He's generated like 10 more lines of different genes of different transgenic lines. And my student now thinks she might even have a line. She's interested in venoms, so she's developing one for venoms and to knock out venoms. So yes, I think it will, but it just takes money. It takes money to pay people and to support them and to keep the lab up and going. And so I think that that's really going to be dependent on that. What hasn't changed in the last few years in terms of the technology is that we haven't gotten any more efficient because we don't know how. I don't think we understand the subtleties of it. But what has changed is just for whatever the background biology is, finding the organisms that are really good at taking up foreign DNA and ones that just don't like it. So what I think is everyone's going to try it on their favorite organism. And some will work okay, and some are going to work amazing. Hydractinia so far has been amazing. Nematostella, ironically, at the Stowers was one of the first to do it, but they've struggled. They've gotten 2 or 3 because I think they're just a really good lab with brute force got it to work the first time. But the organism itself gives them more problems than ours give them. So I think what we're learning in terms of the diversity of CRISPR applications is which ones just work well and which ones don't. And if we figure out why, then that certainly will improve. But the technology itself is so simple, it's hard to improve on. It's just the protein, the guide RNA, and that really hasn't changed much. It's just sort of applying it to different things. I think
So, what's the relationship between the reproductive pathways and the jellyfish pathways and the ability to produce reproductive organs earlier come first?
Okay, yeah.
And then are there pseudogenes left over that you work with?
Right.
Like repair or?
Okay, so you asked actually 2 really, really good questions. It's very science nerdy though, so I think you and I will be the only ones interested in this answer. So his first question was, how is their onset of reproduction, like making the gametes, related to making the jellyfish? And what happened first? That is a broad field of biology to study what's called heterochrony, which is onset of reproduction versus their somatic tissue. And this is a classic example. What happened is they became reproductive before they could develop into a jellyfish. Or they stopped developing into a jellyfish and became reproductive. And I don't know the answer, and that's what you asked. But that's a really good one. And I'm hoping to actually, but it happens a lot in animal evolution. Sort of these different rates of things getting reproductive at a young age, or extending your adult development and delaying reproduction. And there's a lot of evolutionary theory behind it, but this is a classic example of what we call heterochrony. And then, what was that second part of the question? Oh, yeah. So if you lose a jellyfish, the genes will hang out for a while, right? But then you would think that they would go away. And non-functional genes that we can recognize as genes are called pseudogenes. And so that was his question, what happens? So we started screening all the genes, because now we have tons of genomes of different jellyfish, and some have different cnidarians, and some have jellyfish and some don't. But what I didn't mention is some have intermediates. Like, they have jellyfish-like characters, but they don't eat in the water column, or they sometimes detach and sometimes don't. We call those medusoids. They're like intermediates. I can't even believe I found this. I still don't believe it. But we looked at one particular Hox gene that was missing in every single species that didn't have a jellyfish, and was present in every single species that did. And then I found this pseudogene. I'm like, what's going on? It had like a medusoid. It was like right in the middle. So it like fit so neatly, I couldn't even believe it was true. But pseudogenes, I think I just got lucky there, and I don't expect this to happen regularly. But pseudogenes basically are catching intermediate losses. And that was an intermediate loss, and I found a gene marker that matched that. Like, it's lost, it's not really used, and so that was very interesting. Actually, we're going to publish on sort of this pattern of Hox genes and how it relates to medusae. But that's a very good question.
Could CRISPR systems be used to assemble the genome of a synthetic organism de novo?
So what CRISPR systems can do, so you're asking about Jurassic Park type technology. And what CRISPR can do is you basically, I am doing it. So you take sort of an ancestral condition and you try to recapitulate it. So it's not going to assemble whole genomes, but it's going to assemble genes that were missing, or genes that have evolved in the descendant, take a more ancestral genome and try to patch them up. Because you can very specifically now modify those genes. So what did they take in Jurassic Park? Was it a bird or a crocodile? I don't remember.
I had more in mind ground up de novo synthesis of a synthetic organism.
Because it doesn't make genes, it just modifies. So it cuts genes and inserts them.
But you could develop genes for the system to deliver, right?
Sure, absolutely. So what I would say about a synthetic organism is that there's, I mean, even in the simplest of organisms with the smallest of genomes, I mean, if we pare down the bare bones of what it could be, can we patch it together and will CRISPR be useful? Yes. We're definitely not there yet because there's so much unknowns about what make an organism, even a single-celled organism, that we just don't quite understand. But sure, I think in the future that would be a useful technology.
I think one group actually did create something very much like a synthetic organism, and it looked like a very tiny fuzzy snowball.
Oh, okay. Interesting. Did it replicate?
It had everything it needed to do to stay alive. I'm not sure about the replication,
Okay.
or they may have truncated its lifetime.
Okay.
I think that is all.
Okay, thank you.
Thank you for your help.